signing on today. For those of you who don't know me, my name is William Bush, and on behalf of Lisan Wong and I want to thank you for signing on to our first instance of a new multi-institutional seminar series that's focused on next generation researchers that are part of Alzheimer's disease research. And in particular, Lisan and I put this together as part of the Alzheimer's Disease Genetics Consortium and the Collaborative for Alzheimer's Disease Research, two rather large grants that we're both part of and would like to spread to our next generation of researchers. So I guess if this is the next generation, our very own Captain Picard is Dr. Tony Griswold. It's my pleasure to introduce Dr. Griswold. He's a graduate of Notre Dame, went on to earn his PhD in genetics from George Washington University, where he's now assistant professor at the John P. Hussman Institute for Human Genomics at the University of Miami Miller School of Medicine. Many of you may know Dr. Griswold. He has expertise in the use of genomic sequencing, bioinformatics, and so forth to examine neurological disorders, and he's part of multiple funded studies from the National Institute on Aging. Today he's going to present on transcriptomic approaches to Alzheimer's disease in diverse ancestral populations, and so I'll turn it over to Tony.
Awesome. I'm going to try to share my slides. And you see the right slides there?
We do indeed.
Success.
Excellent. Good. So thanks, Will, and thanks everybody for taking the time to listen to me this afternoon. You know, I think Will and I had a discussion about this about a little over a year ago. It was February and it was in Philadelphia. That was the last trip that I was on. And so we initially had this discussion a little over a year ago about setting this up for me to give this presentation. And today it comes to fruition. So here we are, and thanks all for joining. So as the title suggests, I'm going to talk about transcriptomic approaches in Alzheimer's disease, particularly with the focus on individuals of diverse ancestry. And so what I'm going to do is just give kind of a quick background about sort of the reason why we're doing this work. I think I'm going to set the stage a little bit talking about AD genomics, which is probably not necessary for this crowd, but just to kind of get us all on the same page. I'm going to talk about 3 different projects that I've been involved in. So I'm going to try to cram this into about 40 minutes. I think it's possible. Some of this you've heard before, and some of it's relatively new. So I'll talk about a whole blood RNA-seq study that we've done in 80 cases and controls in individuals of both European and African ancestry. I'll talk about looking at RNA editing. So this is sort of an epitranscriptomic phenomena using the same data set as the project above, but looking at it in a slightly different way. And then I'll finish talking about some of our most recent work that I've done looking at single nuclei RNA-seq from the brains of individuals with AD. From both European and African ancestries, and some of the things we see there. And then just finish up with kind of a few sort of final thoughts, you know, that I have, and I think that you know, we can all work together on going forward. And so just to start, we'll talk about Alzheimer's disease demographics. I don't need to describe Alzheimer's disease to this audience, but I think this just puts into context that Alzheimer's disease is a global problem. You know, we recognize this in the US, but really we think about this across the entire population of planet Earth. And it's not unique to us. And so it's estimated that by the year 2050, somewhere around 130 million individuals will have Alzheimer's disease. You know, as populations continue to live longer, that may even be an underestimate. Unless we are all wildly successful in our ultimate goal of trying to reduce the burden of Alzheimer's disease. And so I'd like to see a bubble that we can estimate that number will be smaller at some point soon. We've been looking at the genetics of AD for a long time with a variety of methodologies. And so early on, linkage to familial cases of Alzheimer's disease found the genes APP and the presenilins. We later found ApoE. Peggy and Jonathan's groups, you know, found the ApoE risk allele and showed that individuals with 4/4 had earlier onset and greater severity. Later, genome-wide association studies identified several variants and loci related to these 3 or 4 genes. Whole exome and whole genome sequencing was able to identify rare variants with more impact. And then finally, meta-analysis of large data sets of GWAS has found somewhere on the order of 25 to 30 loci, depending who you ask. We always ask Brian Kunkel, because Kunkel et al. Is a paper that everybody who talks about AG genomics has to show a slide from. And so here I'm showing his Manhattan plot from a GWAS study in 2019 that shows you the roughly 25 or so genome-wide association signals that are associated with AG. And I think that, all of this work has given us a pretty good head start in terms of thinking about the types of genes and maybe the types of pathways that are involved in Alzheimer's disease from a genetics point of view. The problem, somewhat, is the lack of diversity in genetic studies. And so I'm showing you just a kind of a website grab from PBS, and this was from March of 2019, I believe, where genetic research has a white bias and it may be hurting everyone's health. I think that's maybe perhaps a little bit sensationalized, but this came out, and many articles like it came out about the time that this paper was published in Cell, that was looking at the genetic ancestry of individuals that were part of sort of genetic studies in the GWAS catalog. And what you can see immediately is that, close to 80% of all individuals are of European ancestry. Another 10% or so are of Asian, and everybody else is the rest. And I think that, anyone can look at this and see that, that there's a distribution problem here, and that we ought to try our best to even out that pie as much as we can. And to the credit of a lot of people sort of on this call now and our colleagues in the Alzheimer's disease space, we've been thinking about this. And so we've tried through a variety of efforts to increase the diversity in our studies. And so, again, I will pull a Kunkel et al. Paper here. So this was a recently published genome-wide association study in African Americans. And so this is just showing that the Alzheimer's community recognizes that, doing genetic studies across diverse ancestries is important. And these are showing the Manhattan plots. You'll see that they're, instead of being a Manhattan plot, these look more like a, Miami Beach plot or something, where, the peaks are not as high as maybe they would be from the previous GWAS that I showed you. On the top, you just have overall where ApoE is still the dominant signal. And when you condition for ApoE below, some of these other signals start to come out above the pack. Including a couple of known genes like ABCA7, MSX2 loci, and several unique loci that seem to be unique in African Americans. And those are all of your sort of dots and triangles or whatever those are, diamonds in red. And so, why does this look more like Miami Beach than Manhattan when we look at these GWAS? And it's still an underrepresentation of samples. And so we need to continue to expand AD genetics diversity. And I think these numbers sort of point to that, whereas if you look at the number of individuals from the first GWAS study that I showed you, there were close to 93,000, maybe close to 94,000 individuals of European ancestry, whereas the largest African-American GWAS ancestry that I just showed you has roughly 8,000 participants. And if you think about this from the statistical perspective of the amount of power that you have to identify associated variants, there's a big difference when your sample size is an order of magnitude less. And so there needs to continue to be enhancement and increases in diversity in the individuals that we look at in Alzheimer's disease. And this, of course, is continuing. And so just to kind of highlight a few specific projects that I've been involved in, at least on the sort of data analysis side, is expanding this into Hispanic Latino communities. This includes a Puerto Rican initiative and a Amerindian initiative looking in Peru, Cuban Americans. We live in Miami, so that makes a lot of sense. And just a couple of pictures to show, remind people what it used to look like when you could go visit collaborators in Puerto Rico on the left or Peru on the right. And hopefully we can, get those activities back going again soon. these projects and a lot of other initiatives, across a number of different places, all are funneling into things like the Alzheimer's disease sequencing project and the follow-up study, where depending on the day that you ask Brian to show his table, anywhere from 20,000 to 40,000 individuals across a lot of diverse cohorts and ancestry being used for whole genome sequencing, and then the analysis of that feeding into sort of individual groups and then the cadre collectively. And so I think that we're on our way to expanding sort of the number of samples across these ancestries, and with that, the associated increase in power. I really think we're going to be able to identify sort of unique things in these ancestries. And we believe that we will find things. I don't think that this is a futile exercise of just increasing sample size and diversity for the sake of doing so. It's that we know that there can be differences in risk depending on ancestry. And so this is just one example, looking at the APOE4 allele, and that your risk is much more increased if you are of East Asian ancestry compared to if you are an African ancestry or Yoruban. And I think that this really pointedly suggests that there are ancestral effects on your risk. And I think in order to identify these types of effects, we need these bigger sample sizes. APOE is not the only example. There are things like ABCA7, where there are numerous risk alleles across several different ancestries that have been identified. None of them are quite the same. There's a paper by Holly Cookier and colleagues down in Miami, and many of you have worked with her on these projects, looking at a protein coding deletion that seems to be specific to individuals of African ancestry in ABCA7. And so both changes in effect sizes for risk genes, but also specific risk variants can differ between these populations. Again, really underscoring the need to continue to look in these individuals. I'm not going to talk about any of that. Instead, I want to try to take this to kind of what I believe is the next step. We're doing a lot of genetic variant identification and association. I think that's certainly critically important. But most of what we find, and it's not unique to AD, this is across most diseases looking at association, it's unlikely that these are going to be protein coding mutations of high effect. It's much more likely that these genetic variants are going to exert effects by regulating gene expression or other. And so how much RNA is being made? Is that RNA modified? Where is the RNA being expressed? Each of these have important consequences on phenotype, and each can be modulated by specific genetic variants. And so that's kind of one of the major questions that we have. And then really the second part of the question is, do these differ by ancestry? So we know that there are several population-based studies. I just described many of those. And so the question is, can we dig into the functional impact of some of these ancestry, either specific variants or ancestry differential risk variants, and see what their function is at the RNA level. And so the three stories that I'm going to tell you today are really going to address three different parts of those questions. So the first is looking at gene expression. And I think this is perhaps the simplest and highest level way to look at this, which is to ask, are there genes that differ in expression between AD cases and controls? And are those genes different between cases and controls in different ancestries? And so that's the first story that I'll tell you today. The second story is looking at RNA editing. If you don't know what that is, that's fine. I'll explain it shortly. But the idea here is, is RNA editing changed between cases and controls? And is that changed between ancestries? And so I'll talk to you about that. And then finally, we'll ask the question about not how much necessarily of genes, but where are these genes expressed and do they differ between ancestries? And so we're going to use a single cell sequencing approach to look at that. And so these are kind of the three vignettes that I'll tell you today, related but separate. And then again, finish up with some final thoughts of kind of how these things relate and where we can go forward. So I will start with the first study, which is looking at differential expression. And so here I'm just kind of showing you the overall study design. I don't need to talk to you about the diverse populations and why. That's what I've just been talking about for the first 10 minutes. But this is showing you the number of samples that we looked at, roughly 100 cases and controls in individuals of African ancestry or European ancestry. And then we also had individuals from Puerto Rico that are admixed, so they have mixtures of African, European, and Amerindian ancestry. And as a pilot study, we have about 40 cases and controls there. As I'll tell you later, we're continuing to build these data sets much bigger so that we can be a little bit more powered to find things. But as a first pass, I'll talk to you about this. I will say that the RNA expression that we're looking at is from whole blood. And I can tell you, and Will can tell you, that this is a tricky problem, right? That, Alzheimer's disease is clearly a disease of the brain. The problem is that it is very difficult to get brain material, particularly in individuals of diverse ancestries. And so in order to sort of get a first pass at the effect of these genetic variants on RNA expression in this case, but other RNA things, whole blood is really the thing that's most accessible in these individuals. There's quite a lot of data out there that has looked at whether blood and brain share similar expression patterns. Clearly, they're two different tissues, so not all of them, but many, say, QTLs of strong effect have similar effect in both the blood and brain. And so maybe some of these findings will be applicable. If not, maybe this is just purely a biomarker type study looking for gene expressions between individuals. And even that, I think, can give us some interesting data going forward. And then finally, we're going to be looking at both the RNA-seq data for differential expression and pathway analysis, and then follow that up with RNA editing in the next study. And as I've described, we want to really be able to look at this because we believe that there's regulatory changes that are leading to the phenotypes. And so I'm just going to put all of these up rather than animate. We ran relatively simple regression models here, looking at status, correcting for age and sex, and the RNA integrity. For the African individuals, we also included center as a covariate because there was actually a difference in where the cases and controls were recruited from. And so we ran those models in DE-seq and found the number of genes differentially expressed that you see over on the right, about 400 FDR corrected gene expression differences in African or individuals of African or European ancestry, and then a few more in individuals from Puerto Rico. And I show you the lamest Venn diagrams you've ever seen below. I think maybe my initial thought was perhaps that, cases and controls across these ancestries would share a lot of genes, and they share relatively few to none. And so I think initially there was a thought of, well, maybe it's just completely different in, cases and controls in Europeans versus individuals of African ancestry. But I think what is somewhat more sort of comforting in a way is that when we take these and we start to look at pathways that are enriched, we see the same pathways being enriched in the same directions across all three of these groups. So things like immune system processes, RNA processing, response to external stimulus, which is really just another name for immune response. All of those are enriched within these, within each of the populations. And so, I think what that tells us is that while the differences exist between AD cases and controls within each ancestry group, the genes themselves seems to differ. But the underlying pathways and maybe molecular mechanisms seem to converge. And I think that this suggests that, at least looking in whole blood, there seem to be similar underlying mechanisms related to these pathways that may be driven by distinct genes or variants from these different populations. And clearly, I think this is, remains to be investigated more, but I think that this is an interesting first look. And this is a paper that we published last year with a postdoc at the time, Satish and Gary Beecham, who helped drive that. And so some of the details of that study can be found there. I think, once we had this differential expression, we want to go to the next question on sort of my list here, which is to look at RNA editing. And just to talk about that a little bit, RNA editing, it's an enzyme-mediated process where individual bases of mRNA are altered. So these aren't DNA SNPs that are then transcribed. These are actually induced changes in the RNA. It's thought that this was originally part of the innate immune system, such that if you had unrecognized viral RNA, you could modify it in some way so that you could get rid of it. we've now sort of evolved such that this is really a vital function for both neuron development and function, immunoregulation. Really, it's a sort of vital process. If you knock out the enzymes that are involved in RNA editing, those are lethal. And there are a lot of ways that these edits can change things. So you can introduce coding changes, you can change splice sites, you can create microRNA bindings, you can change the stability, you can do all kinds of things to the RNA. And so this is just a cartoon of how this works in sort of a canonical way, where if your mRNA is sort of here from 5' to 3', and you have a complementary thing, you can get a sort of a hairpin structure. And in these double-stranded RNA regions, there's an enzyme called ADAR that will recognize this and will sometimes modify the A into what is actually an inosine. And then we read that off on sequencing as a G. So what an edited site looks like is a change from an A to a G. And I'll just note that almost all, sorry, I'm getting a phone call apparently. I'd like to note that all of the work here was done by a really talented graduate student, MD, PhD student, Olivia Gardner, and this is her picture. And we published this paper maybe a little bit over a year or so ago. She's really good. I miss her because the work that she did was really strong. And so just to give you an example of what might RNA editing be doing. And so this is just one example. It's a pretty famous example. It's published in this paper in 2012. It's one that a lot of people point to. And this is looking at the GLU-A2 gene and one specific site called the QR site. And so if you have unedited RNA for this particular channel, the channel is impermeable, right? And so this calcium is not able to go through. Once you have RNA editing that occurs, and again, this is not a DNA mutation that changes from Q to R, this is induced on the RNA itself, this channel becomes permeable. And so this is really then a tunable way that you can turn this channel on or off by modulating the actual RNA. Doesn't seem the most efficient design by nature, but is a pretty well documented example. And there are actually relatively few transcriptome-wide studies where people have looked at RNA editing. And as far as I'm aware, ours may be the only one that's done this in a disease-specific way, and certainly the only one that's looked at individuals of different ancestries. And so we really thought that there was a gap here that we could begin to fill. Right. And so just to give you an idea of how do we detect editing. So we're going to go back to the raw data for our RNA-seq. We're going to look at every individual position. So if you think of these green bars here as sequencing reads, and at some position here, there may be, the A is coded in the DNA, right? But every now and again, you'll get editing that will give you 2 Gs. And so if we were to see this site, we would call this site 20% edited, right? And then a different individual, you might look at all of the reads and, say, okay, well, now there are 4 Gs, so this person is 40% edited. And you can imagine that somebody would be 80% or 100% or whatever it may be. The key here, of course, is that there's a lot of filtering that goes on in terms of quality to make sure we're not looking at sequencing errors, to do our best to make sure we're not looking at underlying rare SNPs and things of that nature. And so this is kind of the output of how RNA editing is detected. And so, again, this is just sort of what I think these slides were out of order, but that's fine. So again, we want to look at this epitranscriptomic analysis to look at downstream effects of AD-associated genetic variants, that this might be related to pathogenesis, and it's really understudied. And so we sort of wanted to fill that gap. And so this is just to show that there were a few less samples here that we were able to push through because of some QC issues, but essentially 100 cases and controls from our African and European ancestry individuals. We used a tool called Reddit Tools that will perform those calculations that I just described, looking at the G versus A counts. We do quite a lot of data filtering, looking at position in the reads, quality of the base. We filter out, any SNP that's identified in dbSNP. And so we have a pretty stringent filtering criteria. We've done differential editing. So we just ask whether There's a difference in the frequency of editing between cases and controls at every site we identify. And then we take a look at what genes those are that are edited to see if there's any relationship among them using a pathway enrichment analysis. And so just a very high level picture of the data, we identify about 45,000 total editing events across the transcriptome in all of our individuals. 87% are these A to I type variations. We would expect that. Most people who have looked at RNA editing find, anywhere from 85 to 95% are these canonical changes. And so we're well within that range. 83% of these have already been identified in some database of RNA editing. So there are two large ones, one called RADAR, one called DARNed. They're fun names. And they are sort of catalogs of RNA editing sites, and most of ours overlap them. And also most of these occur in ALU repeats. And so given the sort of nature of those hairpins that I described a few slides ago, editing events are really common in ALUs. So particularly ALUs that exist in long 3' UTRs or in intronic regions, that tends to be where we see most of our RNA editing. And so again, all of this matches up with sort of the existing literature for RNA editing mechanisms. And so we're kind of right on track with what we expect to see there. And when we look at differential editing, we see a lot. So we see about 500 positions in 273 genes in our African-American individuals that differ in RNA editing, 700 positions in about 300 genes in our non-Hispanic white Europeans, and about 100 genes that were differentially edited across both in cases and controls. I'm just showing a heat map on the bottom just to show you that this isn't kind of some phenomena where there's one individual who's really driving all of the difference. But these things that are differentially edited, in green are positions with lower editing, and the exact opposite. Green has higher editing, red has lower editing. And you can kind of see these clustering of your cases and controls here, and it's not perfect. But if you squint your eyes hard enough, you can see what looks like a pretty clear pattern here of your cases controls with differential editing. And so what we did is we wanted to look at these genes. So we looked at those, roughly 300 or so unique in African American, the 600 or so unique in the non-Hispanic white Europeans, and then the 94 that were differential in both. And again, just ask the question, what can we say about those genes? Is there anything sort of similar about them? And there's sort of a lot similar about them. And so, here I've kind of colored these by sort of ballpark pathway. There's a lot of sort of cell signaling type things in yellow, phagocytosis and endocytosis in blue, and then sort of cell migration type neutrophil activation in green. And so, again, we see that the different genes seem to differ. The pathways, again, seem to converge. So this sort of matches with some of the message that we saw from our differential expression as well. And, the last thing that we wanted to do is sort of ask a question about, well, we've looked at this in kind of the really broad sense of what genes are they, but what is the effect of any one of these variants? Can they really have biological meaning? And so this is just one example. I don't know anything about this gene called TAP-BP. But I think that this sort of shows the possibility of how editing can change something. And so I'm showing you two sashimi plots here. So this is looking at the raw RNA data. So along the bottom are your genes, where these dark blue bars are your exons. These kind of, hashed lines are introns. And sort of the peaks up here represent read coverage, right? And so, this kind of this is part of the 3' UTR of this gene. Has a lot of coverage, and then each of these exons here have little mounds of coverage. And then these loops represent splice junctions, right? And so here you have, a splice junction that goes from this exon all the way to the last exon, and that's sort of what you would expect to see. In individuals that have RNA editing at this specific site, so chromosome 6 at this position, you have editing that exists here. And what that does is it stabilizes a sort of weak splice site that already exists in this ALU. And so what happens is you now get splicing events between this ALU and the last exon, and between the other end of the ALU and the previous exon. And so this actually results in alloexonization, where now you have, this allo isn't part of the thing that's spliced out, but it's actually built now into the final transcript. Now, in this case, it's in the 3' UTR, and so maybe it's not incredibly functional. But I think that this is an interesting phenomenon, and we're now continuing to look at this, sort of genome-wide, transcriptome-wide, looking for other sort of examples where this RNA editing has a really demonstrable effect, either in splicing, and we're also combining this back with the expression data. And so I think that there's kind of some really interesting functional things that can still be mined from here. And we even took it one step further, right? So we looked at the differential overall between cases and controls. We've started to look at the individual effects of editing sites. And the last thing that sort of Olivia really wanted to look at was whether we could identify DNA variants that modulate this effect, right? So we can see that there is difference, and then in the same way that gene expression can be modified by expression QTLs, we wanted to investigate what we're calling editing QTLs or EDQTLs. And so this is just a very simple cartoon that's kind of giving you the lay of the land, which is that you can have some SNP somewhere in the genome. You can have RNA editing at some level, and then depending on the genotype of that SNP, the level of RNA editing differs. And so you can imagine a scenario where if you have a 0-0 genotype, whatever that is, maybe your editing is 20%. And if you have a 0-1, maybe it's 50%. And if you're homozygous for whatever that SNP is, maybe you have 80%. And we are somewhat underpowered to identify these. We only have about 100 individuals, about 200 individuals in each sort of ancestry, and even less so if we split those up by disease status. But we thought that this was an interesting question to start to look at. And so what we wanted to know is whether there were variants in AD-associated loci that act as editing QTLs, which I think would be an interesting tie-in to give some functionality to those variants, and also to see whether there were editing QTLs that could explain some of these differentially edited sites that I just described previously. And so that's what we set out to do. And so again, this is a super blurry slide apparently, but we are using the same data set. These individuals all have genotyping on the global screening array, so we're looking at about 640,000 or so markers. Many of them have whole genome sequencing, and so this may be an analysis that we go back to look for other variants that might be doing this, but again, this is kind of a first pass. We're using the RNA editing that I just described, so these same 45,000 sites. And then we use a hurdle model because there's a lot of zero inflation here to look for editing QTLs around these editing sites, and then adjusting for multiple testing and pulling out those with significant FDRs. So this is just to summarize in these pastel colors. So we see roughly 2,000 editing QTLs among our African cohort and around 3,500 in our Europeans. And then showing you that about a third of those, depending on which population, overlap between them. And on the right, I'm just showing you how far away from the editing site do these SNPs exist? And they tend to be very close. That's not too surprising. Some of the sort of proposed methods for how an editing QTL may work is by slightly changing the two-dimensional structure of these hairpins where editing occurs. And so you would expect that your SNPs would be relatively close to your editing site, and that's exactly what we observe in this graph here. And so what I wanted to do is just show you a few examples of what these things look like. So we have, as I showed above, about 1,100 of these that have a shared effect. And so what you can see on this graph on the right is just that in both your African and European individuals, when you have, the 0 genotype for this particular SNP, you have roughly, 60% editing. When you go down to a genotype of being a heterozygous at this SNP, it's roughly 20%. And then though there are few individuals, if you're homozygous, you actually have the smallest amount of editing. And so this is showing a shared effect where both ancestries have the same effect, no matter their genotype here. There are other times where we have ancestry-specific effects. So the difference is a little bit more subtle here, but if you look in blue at your European population, what you'll see is that your editing increases as you change your genotype, whereas your Africans stay roughly exactly the same at 50%. There's no increase there. And so we see several of these, over 2,500, where in one ancestry, the allele doesn't seem to change editing at all, whereas in the other, you do see this. Sometimes you see a reverse effect. It's not as common, so we only found 100 of these, where you'll see that depending on your genotype here, the African rate of editing increases, where it seems to decrease in the Europeans. And again, there's only 100 of these, but I think that they're pretty interesting. And then we also have several ancestry-specific alleles, right? These are things that have frequencies that are too low in one or the other populations to even measure for being a QTL, but they do turn out to be a QTL in the other populations. So we do have a number of those as well. We wanted to see whether these were related to AD loci that have already been identified. So we looked at the signals from the two Kunkel papers. We looked at LD blocks to pull out all variants on the GSA that were there. And then within those, look for EDQTLs. There were relatively few, other than this really sort of strange phenomena that happens in the HLA region, where there are actually quite a lot of editing QTLs, and I suspect that might be some sort of data artifact that we're still looking at. But here you'll see that there are a few specific AD loci where there are editing QTLs, and so here's one that's shared between both African and European backgrounds in the SORL1 gene. And so these are things that we're sort of following up on to see, really what their effects are. And then finally looking at editing QTLs for differentially edited sites. And so there were 148 sites that had underlying editing QTLs. And so I'm just kind of showing an interesting example down here. Again, this is from the HLA, so take it with a grain of salt. But what you'll see here is that, your cases and controls in sort of pink and blue have a big difference in genotype frequency, right? So your cases tend to have a lot more of the 0 genotype, and your controls tend to have more of the 2 genotype in this case. And so this was an example where there was a differential editing that was driven by this genotype, but that was also related to case control status because of this difference. And so I think this is really getting to this idea that, variants that, this might just be sort of a nominally significant variant that may exist on a GWAS or some type of other study, but it may really have a real functional effect that may be worth looking into. And so, again, I think something that's sort of interesting. And so just to summarize this somewhat long middle piece of my talk, like gene expression, we see that genes with differential RNA editing are enriched in immune, inflammation, endocytosis pathways that we've seen before. We've found some editing QTLs that explain some of these differences between cases and controls. And while we don't see a lot of evidence for editing within AD loci, I think further investigation is necessary here, looking at both whole genome sequencing so that our catalog of variants is bigger, and then obviously having bigger sample sizes to go all the way through this. And so all of that being said, we have just sent out to co-authors, many of whom are on this call, a version of a manuscript that summarizes all of this editing QTL work. And so this is a nice kick in the butt to go take a read of that and send any comments back our way, because I think that this is really interesting and something that we should follow up on. And so I'm going to move on then to the last thing that I want to talk about, which is kind of a different data set and sort of a new and interesting thing. And so this is looking at cell type specific expression and whether there are differences in AD between ancestries. And so this is a single nucleus RNA-seq study. And so again, I've already mentioned this, but this is just to show again that the risk for the APOE4 carriers differs depending on your ancestry. So those with East Asian have the highest risk, Europeans somewhere in the middle, and then Africans with the lowest risk. And this was sort of shown somewhat recently by Fareed Berjabli in our group, that this wasn't just related to sort of global ancestry of individuals, but really related to local ancestry around the ApoE gene itself. And so we've sort of had this hypothesis in question about looking at the local ancestry region around ApoE in these individuals of diverse ancestry, and asking whether we can identify specific genes, specific cell types that might explain some of this AD risk variation. So this is just to kind of give you kind of a high level view of the study as it was at the point when these slides were made. We actually have a few more samples that we've looked at. The major conclusions haven't changed. And so first we screened through the NAC, the National Alzheimer's Coordinating Center, to identify all of the sort of brain material that we could identify that were AD, homozygous for ApoE4, and that were primarily of African ancestry. We then screened and confirmed the local ancestry for those individuals around ApoE. And what we ended up with at the time of this analysis was 7 individuals of European local ancestry, 4 of African local ancestry. We looked in the frontal cortex of the brain, frontal cortex of the brain. We did single nucleus RNA-seq from using the 10x Genomics Chromium, and then did some sort of informatics with Cell Ranger, and then the Seurat pipeline, which is the data that I'm going to show you in the next few slides. So the first thing that we did was just to sort of cluster the cells and ask what they look like. And so again, this is all tunable depending on your parameters of analysis. But in this case, I'm showing you 32 defined clusters. So every dot on this plot represents a cell. And sort of the closer together any two cells are, the more similar their expression pattern overall is. And so we get about 32 clusters here. And when you dig a little bit deeper into what genes are defining these clusters, we're actually able to show 8 distinct cell types. And so these shouldn't be surprising given that we're looking in the brain. But we're sort of able to sort of, take this sort of mishmash, and these are all excitatory neurons. If we really dig deeper, we can start to dissect out the different types of excitatory neurons. And so a lot of our analyses, tried to get down to a really sort of granular resolution to see whether there were very specific cell types and clusters that had differences. But this just kind of gives you the overall picture of what we see. And again, I don't know that much is very surprising there. I think what was really interesting to us was, oh my, I missed a slide. Bummer. Okay, apologies. So one of the things that we were really interested in, and you can't see it here, is this particular cluster that's floating here. What we identified is that ApoE gene expression was much higher overall in European brains than in African brains, in both astrocytes and microglia, and in actually several other cell types as well. But what was really interesting was this sort of what's called cluster 20 here, but is also this astrocytic cluster. It was only present in individuals of European background and expressed a huge amount of ApoE. And so what we wanted to do is first just confirm that these were astrocytes. And so that's what I'm showing you here, is just that all of the marker genes, or many of the marker genes from that cluster, show that these are astrocytes. And what was really interesting to us is that not only were these astrocytes, but they looked like they were overexpressing quite a lot of markers for reactive astrocytes. So here are just 3 examples of sort of pan-reactive astrocyte markers that were much higher in that small cluster of astrocytes relative to the other. And that these were A1-like, so that these were the neuroinflammation type reactive astrocytes. And so that's all I'm showing you here is a few of those markers that suggested this. And so just to summarize kind of this whole single nuclei study, we think that this is probably the only one that's looked at this in this particular way, looking at African and European ancestry, specifically in ApoE4 patients. And what this really suggests is at least that some of the risk of AD related to being European relative to African may be due to this ApoE expression increase, and possibly related to this increase in reactive astrocytes. And so we've continued to add samples onto this study. I think we're now at roughly, I think it's 17 total samples, more or less split between Europeans and Africans. And these results continue to be robust. And so this wasn't just a matter of we found a few samples where we had this observation. We've actually seen this as we continue to build our sample cohort. And the data that I just showed you was published very recently in Alzheimer's and Dementia. And just to show that I worked really closely with a postdoc in the lab, Katrina Stellis, who did a lot of the sort of bench work for the single cell. And again, I think that it's worth it to go look at this paper because I think it's really quite interesting. Sorry. Okay, so I am 45 minutes in, and so I think I'm pretty good on time. So it's a good time to throw up some conclusions. And so, ongoing genomics, and functional genomic studies like the ones that I've described here, they all point to there being different underlying risk genes and variants for AD. And I think that there's no surprise there, and I just kind of want to, continue to stress that we really need to have all of not only just our genetics, but also our functional genomics work be done in these other populations as those effects may not be necessarily consistent across. This idea of moving into sort of single cell work and looking for cell specific responses is going to be even more critical to give context to some of these studies. And then hoping that we can identify these common molecular pathways that emerged so that anything that we do in terms of either drug discovery or therapeutic development is really generalizable, that we're not targeting just European genes, pathways, and variants, but that things can really be applied broadly. And then just finally, some looking ahead. So I'm always thinking about, when I have to give this talk the next time, what are the things I'll be thinking about? And so the first is sample size. So we have ongoing RNA-seq for several more individuals of diverse populations. We're going to continue to add on to the data sets that I presented here. It's going to increase our power. We're going to identify more. Diversity. I wasn't very creative in making these bullet points, but I think that this just underscores that we need to really be looking in not just African and Europeans, as I described here, but individuals from Amerindian, Asian ancestries, admixed populations. I think, we really need to be inclusive of what we look at here. We've started to look at, I have a talk that I'll be presenting at AAIC this year, looking at expression QTLs based on some of the data that I've shown. And so we will see some of that soon. We've started to think about epigenomic regulation. So I've shown you some of the expression differences, in say the local ancestry, depending on European or African, but what's driving that? And so we've started to think about this from an epigenomic context as well. And so that's to be determined. And then there's also a focus of thinking about, once we have evidence for any of these things, how are we going to test them? So thinking about things like massive reporter assays, cellular functional models, animal modeling, these types of things that follow on is going to be something of importance. And then for any of the effects described above, how can we modulate these? So looking at things like genome editing or drug screens from cell lines or whatever they may be, that's perhaps the longest term, but I think that's the ultimate goal is how do we take, genomics, functional genomics, cellular outcomes, and then really think about how we can modulate those in some way. And so finally, I just want to thank a lot of people. So Peggy, Perchek Vance, and Jeff Vance, who are the PIs for the grants down in the bottom right that really paid for most of this work. And then some of the other collaborators that I have, the clinical teams at Miami and with collaborators at Wake and Case and really from all over, people in our sequencing cores that have helped us out. And thank you guys for listening. And hopefully I didn't run too long over. And then again, I'm happy to take any questions that anybody may have over the last minute or so.
Tony, I sent around an article that Eliezer Maslia sent to me Monday or Tuesday, I forget. Have a look at that because I think there's pieces of your puzzle in there.
Great.
It has to do with ApoE. Okay. And it has to do with selection of cell pathways for death, all having to do with a major histocompatibility component. So I think everybody is kind of converging on this all at the same time.
Yeah, I think that's a good thing. I mean, I'd like to hope that all of us can converge at some point. That's I think our ultimate goal. So thank you. I'll definitely take a look at it.
There was one question in the chat about divergent significant differentially expressed genes across populations with different ancestries. Do you really think these are the result of different effect sizes? And presuming the alternate would be just, you know, differences in ascertainment or sample size or something like that, right?
Well, yeah, it was me, Oscar. Hi. Thank you. Great talk. So I was thinking in context of the different effects of APOE, right? And with reduced sample size, whether you found, right, similar pathways. So within those pathways, might be the case that you have different effects in different populations?
I mean, I think, you know, as Will phrased it, it could be any or all of these possibilities. And I think that, you know, when I look at, let me think of the right way to phrase. So if we just look at the genes that are differentially expressed within each population, they tend to be unique, right? If we take all of the samples together, regardless of ancestry, we don't necessarily get the sum of the two populations separately, right? So there is some interaction there that's happening between, you know, just being a case or control. There are a few genes that are always going to be different, and combining the two ancestries actually brought those up towards the top. And so it's actually a third analysis that we did in the paper that looked at kind of what I'd call, I guess, maybe the joint analysis of all of the data together. Those pathways for the joint analysis really look pretty similar to those that are separate. And so I think, you know, what we're seeing, at least what I'm seeing, is no matter how I slice the data into ancestry specific or sort of the joint of everybody together, I'm kind of seeing these same types of pathways emerge. I'm not sure that exactly answers your question. I think that, you know, this idea related to effect size, yeah, sure. I think as our sample sizes grow, that kind of Venn diagram may converge a little bit more than it has now. You know, we may be, you know, if in one ancestry the difference is just below our threshold, you know, we may not, you know, be capturing that. And so I think that, you know, as we continue to add more data, we'll be able to get at that a little more directly.
Okay, yeah, but my question was more related, like, basically, there's no pathway that it's hitting worse any of these populations. That's what they.
So I guess a different question of whether there are pathways unique in either population.
No, not the strength. So basically, do you, in the same way that you see differences of the ratios in APOE, do you see those differences in the pathways in general? That you can say that whatever pathway is more European and some other pathway it's affecting more deeply people with African ancestry, for example?
So I do think we see that. I don't know that I'm prepared to give the actual specifics to that, but we do see some pathways that are more, I don't want to, maybe more enriched is the right way to say it. So that maybe there are more genes contributing to the pathways in one or the other ancestry. So we do see that. We do see some pathways that look like they are stronger, let's say, in one versus the other, but that when you know, so that they aren't identically named, say, Go terms, for example, right? That, you know, one may be called inflammation response, and the other one may be, you know, TGF beta response or something. Many of the genes are different, but if you look at the overall pathways, they're similar. So I think the answer to your question is, yes, there are some pathways that are more strongly enriched in one ancestry versus the other, but that if you looked at kind of the higher level parent pathways, they're actually quite similar.
Thank you.
Yeah.
Jonathan, did you have a question?
I did, and hopefully it's quick. So I was interested in the single cell analyses that you did with the ApoE44s.
Yeah.
By local ancestry. Have you thought about, in a somewhat different context, have you thought about, and then maybe there's no tissue available, looking at individual cases and controls who are ApoE44? So finding a few ApoE44, older controls and looking to see if you see that same kind of thing, or if there's something different going on there.
Yeah, so we certainly have thought about it. So there's kind of two big thoughts that we've had. One is certainly looking at cases versus control. The other one is looking at individuals of Asian ancestry that are E4s to see if we see even a bigger sort of amplification of what we've seen. I think that the tissue is probably out there. It's just a matter of tracking it down and being able to use it. Especially in single cell data, these repositories are getting bigger and bigger and bigger. And so it's probably just, in some ways we may be able to mine what's already existing. I know a lot of people maybe on this call are generating a lot of single cell data, just data generation. And then it's a matter of trying to go in there and mine that with that type of hypothesis in mind. And so I think it's a great question. I think it's exactly the direction we want to go, is now try to expand. I think, twofold. One is looking at, can we explain this effect, looking at things like epigenomics and things like that. And the other is, how does this look in case control or Asian ancestries? And I think those are things that we're still building on.
Yep, thanks.
Tony, there was a similar question in the chat about tissue specificity of the RNA editing. What do you think about that?
Yeah, so it's a really good question. It's one that we haven't, in our group specifically, we haven't done a lot of bulk RNA sequencing at this point. And so we can't look at least using the 10x technology, we can't look at editing in single cell. And we've done some bulk RNA from brain. We've done really, but we're talking sample sizes on the order of like 8. And so, so we've done some of that. I can tell you that many of the sites that we see are the same, right? So I think the way that I imagine RNA editing sites looking is that there are some that are really consistently edited. And you're going to see those in almost every tissue that you look at. There are certainly those that are unique to tissue. So there is certainly a tissue specific effect. And we are, I think we're sort of trying to think about ways that we can look at things like, what I haven't done and what Olivia had proposed to do and we didn't get around to before she graduated, was to look in things like the Ross map data and look at RNA editing and see what's there. And it's still kind of on the agenda. Just kind of died on the vine when she decided that she had to go back to med school. But I think it's a good question. I think we will see some of the same. I believe we will see differences between cases and controls. But we haven't done that experiment yet.
Well, we're at the end of the hour. I think we'll call it there. I know Dotto had a question in the chat. Tony, you might take a look at that and maybe
yeah.
Offline.
The answer is only those expressed in blood. That's the answer.
Well, thank you so much, Tony, for a great presentation. Thank you all for attending today. We'll reconvene probably in about 3 months. I believe our next speaker is going to be Alessandra Chessi from University of Pennsylvania to talk about some of her work on reporter assays. And so we hope you'll join us again then. Thanks.
Thanks, everybody.